आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज।
Celebrating our Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Radioactive Community Radio, Bengaluru.
एक नई दुनिया की आवाज आपकी आवाज, विज्ञान की आवाज, की आवाज, की आवाज।
Latest information on STEM popularization.
Mutations, the navigators in the evolutionary journey. A team of scientists from the National Center for Biological Sciences, Bengaluru, have discovered that mutations not only provide genetic variations but also actively influence evolution, aiding organisms in adapting to new environments. Their study on E. coli revealed that altering mutations biases can lead to greater number of beneficial mutations, providing new insights into evolution. Mutations are changes in the DNA sequence of an organism. They are the source of genetic variation, but changes brought about by mutations are often regarded as random. However, scientists from the National Center for Biological Sciences, Bengaluru, and Western University, Canada, have made a significant discovery regarding the role of mutations in evolution. The research has revealed that mutations not only provide the fundamental material for evolution, but also actively influence the types of mutations that occur. This can aid organisms in adapting to new environments and drive the evolutionary process. Deepa Agase, Associate Professor NCBS Bengaluru, who is also one of the corresponding authors of the study, said, "Our research lab has always been studying how mutations contribute to the adaptations for a while. When some kinds of mutations are favored over the others, it is termed as a mutation bias. We started this project to see if changing the mutation bias affects an organism's ability to adapt and how bacterial genomes evolve. Previous studies have looked at the impact of the intentionally causing certain types of mutations more than others. The researchers conducted this study on a bacterium known as Escherichia coli. They designed experiments and used computer simulations, drawing on their knowledge of how this bacterium has evolved over time. They worked with two types of E. coli cells, the normal cells, referred to as wild type or WT, and a modified version which was created by removing a gene responsible for DNA repair. The researchers associated colonies from both strains and performed a technique called whole genome sequence on them. This method allowed them to determine the complete DNA sequence of the E. coli genome, which consists of about 4 to 5 million building blocks called nucleotides.
Now, our next segment is Analyst in STEM. Today, we are here with Professor Amitab Joshi, Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. He was trained as a geneticist, but switched to evolutionary biology for his doctoral work.
I do basically both experimental and theoretical work. All our experimental work comes under the broad framework of experimental evolution in ecology, which is to say that we address some fundamental questions in evolutionary biology as well as in population ecology using laboratory systems of Drosophila or fruit flies as the model system. So we do long-term multi-generation experiments and track evolutionary change or population growth changes in real time. For example, the longest running experiment at the moment in our lab is more than 900 fruit fly generations long.
Okay.
And we have been seeing how these flies evolve to become faster developing, because that is the selection pressure we post on them. Every generation we select only the fastest developing 15 to 20% individuals to breed for the next generation.
Okay.
And over hundreds of generations, these flies are now completing their development from egg to adult more than 60% faster than their ancestors would have.
Okay.
So almost 40-45% reduction in the duration of development is there. Then we use these populations to study various other things. For example, if you have evolved to become such a fast developing fly, what else has changed?
Okay.
How that is achieved? What all is changing? What other characteristics are contributing to this change? Similarly, we have very long-term studies going on in the area of adaptation to crowding.
Okay.
So how organisms will evolve if they are chronically subjected to highly crowded conditions. What kind of changes they evolve in their life history, in their physiology, in their behavior, etc. So that is another area in which we have been doing a lot of work. And theoretically we do some work on the basic conceptual foundations of evolutionary theory, such as proposing better ways of defining fundamental things like natural selection or Darwinian fitness and so on, how we should conceive them, what is the role of heredity in selection, is heredity always through genes or can there be non-genic heredity also. So some of those issues. Also that work which I do is in collaboration with a colleague here, Professor Vidya, and two of our alumni, Professor Prasad in ISER Mohali and Professor Sutirth Dey in ISER Pune. So that conceptual work, four of us, we do as a group which we call as Foundations of Genetics and Evolution group.
Sir, how is your research useful for a common person or in the field of science?
So our research is basic research. It is not aimed at specifically solving some immediate practical issue that is presenting before society. So in that sense it is not directly relevant to the average citizen. But it is contributing to our understanding of the process of evolution, how evolutionary change happens, how genetics and ecology interact to produce the particular patterns of evolutionary change that happen. And this basic knowledge sometime in the future may lead to interesting applications. For example, evolutionary biology is typically considered a field that is not particularly applied. But today if you see, in cancer biology, evolutionary thinking is being applied because people have realized that a cancer that is growing in your body can be treated as an evolutionary problem. Your body is the environment. Cancer is a population of cells which is evolving. And that is why actually often tumors will develop resistance to chemotherapy, etc., because they have evolved that resistance. So today evolutionary principles are being applied, for example, in cancer biology to better manage the cancers, devise better interventions. COVID we have all been living through last 4 years. You are hearing all this talk about variants changing in frequency. That is all evolutionary process at work. So if we contribute to better understanding of fundamental evolutionary processes and phenomena, tomorrow it may have useful impact on some practical issues also, but that is not the purpose for which we are doing the research.
Sir. Sir, can you give some insights on the history and the present development and also the future scope of this research?
Okay, so historically, modern day experimental evolution, by which we mean the use of large replicated sets of populations which will be tracked over tens or hundreds of generations in the laboratory to study the evolutionary process as it is happening in these populations. See, traditionally it is believed that evolutionary process you have to study fossils and other things because we have not seen what happened. This is saying we will set up evolutionary experiments. We have a population, we present it with a challenge, then we track it in real time over many generations and see how it evolves in response to that challenge. So this kind of experimental evolution really began around the late 1970s with the work of Professor Lawrence Mueller in the US and subsequently Professor Michael Rose. In that time he was working in the UK, later on he moved to US. They started this kind of work with fruit flies, and subsequently experimental evolution with bacterial systems was pioneered in late 1980s by Professor Richard Lenski, also in the US. During my PhD and postdoc days, I had actually worked with both Professor Larry Mueller and Professor Michael Rose. And when I came back here, I more or less introduced experimental evolution in India. Prior to this, there was one place where experimental evolution was done, although that term was not so popular that time, but Professor Ranganath and his group, starting from 1970s in Mysore University, they were doing experimental evolution work with fruit flies. Very interesting work on hybridization followed by race formation and incipient speciation. After that, basically now I have been here since 1996, so quite a few of my former PhD students are now faculty in various institutions in India, and many of them are continuing to do experimental evolution research, but on questions that are little bit different. So approach is the same. So today in India we have about more than 10 or 12 labs that do experimental evolution research. Out of that, about 12 labs, roughly 10 of them are people who are either my former students or were postdocs with me. Couple of them are independently coming with training from different places and they have set up their labs. So today we have a good, actually a very strong experimental evolution group in India. So these labs constitute about 50% of all research groups worldwide in the area of experimental evolution. India is about 50% of the total labs in the world.
Sir, considering the research work, in terms of ranking, where does India stand?
Well, as I said, we contribute about 50% of the labs, research groups in the world that use long-term experimental evolution studies. That's a pretty high number. I mean, think about it, how big is the world and how big is India, and we are 50% of the labs. In the particular area of adaptations to crowding, which is often technically referred to as density dependent selection, that is, what kind of selection the population will face depends on what kind of density it is experiencing. In the area of density dependent selection, for the last 22 years or so, we are actually the number one lab in the world. In the area of how population stability evolves, that is to say, whether a population fluctuates a lot in its numbers or it maintains relatively steady numbers, and whether this kind of stability in terms of maintaining steady numbers and not going extinct, how does that stability evolve? There used to be a lot of theoretical work on this in the 1980s and 1990s. Many different explanations were proposed why this kind of stability evolves. Since the year 2000 till date, there have been 7 experimental studies on this field. 6 of them are from our lab in JNC Bangalore. One of those studies, which was published in 2003, was actually the first experimental validation of any of the theories for the evolution of population stability. And that particular research recently was included among 65 pieces of research that are considered as conceptual breakthroughs in evolutionary ecology in a book of that name. In 2019, there was a book published by Elsevier, authored by Professor Larry Muller, which is called Conceptual Breakthroughs in Evolutionary Ecology. So in that book, he discusses 65 studies, starting from Charles Darwin 1859 till about 2010 or 2015. So totally 65 studies are included. The 2003 study from our lab is one of those 65. It's the only study from a non-Western country that is included in this list. So in these areas of evolution of stability, adaptations to crowding in fruit flies, density-dependent selection, we are actually the number one lab in the world for maybe last 15-20 years.
Sir, can you brief about the contemporaries?
So as I said, there are quite a few people doing experimental evolution in India. Now the questions they work on may vary, but the approach is the same, that we will set up the system, watch it evolve in real time. Rather than speculate what might have happened in the past, we will see what happens.
Okay.
So for example, a former student of mine, Professor N.G. Prasad, is in IISER Mohali. They use experimental evolution to study issues like sexual selection. That is, selection acting not through differences in survival, but differences in how good the individual is at obtaining mates. And sexual conflict between males and females. This is conflict at the genomic level, not physical conflict. And Professor Prasad's lab right now is pretty much among the top 3, 4, 5 labs in the world in that area. They have, for the first time in a common experimental system, they have actually tested all the predictions of sexual selection and sexual conflict theory together. Earlier, they were tested piecemeal. One guy tests one prediction, somebody else with a different system, different species, tests another prediction. Prasad's group has tested all the intertwined predictions in one experimental system. It's a very superb piece of work. Professor Sutirth Day is in IISER Pune. He does experimental evolution both with bacterial systems and with fruit fly system. With fruit fly system, they have done some of the first studies in the world on how the tendency or propensity to disperse evolves. This is very important today because of changing climatic patterns and changing anthropogenic disturbances of environment. Lot of species, their ranges are shifting. They are moving to new areas, they are leaving their old areas, and this has all kinds of implications even for new diseases spreading. So they have actually done some of the first studies to show how this propensity to disperse away from your native place, how this property evolves. What are the constraints on its evolution? What are the associated traits that help these populations evolve? So this is again a very unique piece of work in context of the international scene. And with bacteria, they did also a very, very unique study on how organisms adapt to randomly fluctuating environments. See, most of the time when we do experimental evolution, we have a constant environment. We provide some environmental challenge, which is constant. If it is a crowding, it is always crowding. Then we see how the population evolves in response to that. What they did was that they challenged bacterial populations with an environment that every generation is randomly changing with respect to pH or temperature or some other environmental axis, and then ask how do the populations evolve to adapt to this fluctuating environment, and whether the traits, the characteristics that evolve are different in this case compared to if you were to present the population with one constant environmental challenge at a time. And the quick answer is the traits are actually quite different. So if you adapt to randomly fluctuating pH, temperature, etc., the traits that evolve are different from what happens if you adapt to a fixed high pH or a fixed low pH or a fixed high temperature or a fixed load. And most interestingly, what they found was that even though the fluctuating environment in these experiments did not involve antibiotics of any kind, nevertheless, the populations that adapted to the fluctuating environmental conditions also evolved multiple antibiotic resistance as a byproduct of more general mechanisms of dealing with stress. This is again quite fascinating because, as you know, multi-drug antibiotic resistance is actually a practical problem of considerable concern today. And this suggests that antibiotic resistance could, in principle, evolve even without indiscriminate use of antibiotics.
Okay.
Which is a frightening prospect, quite honestly. So these two groups are perhaps the most well-known in India.
Okay.
There is also Dr. Shampa Ghosh in Kalinga Institute of Technology in Bhubaneswar.
Okay.
She works more on what you might call evolutionary developmental biology and the evolution of body sizes and growth rates. Professor Mallikarjun Shakarat is in Delhi University Zoology Department. His group works on life history evolution, evolution of aging patterns and stuff like that. Again, all of these are using experimental evolution approaches.
Okay.
Professor Subhash Rajpurohit is in Ahmedabad University. He is, by the way, not a former associate or student of mine.
Okay.
He does work particularly on adaptation of populations to changing climatic conditions. And then there is Dr. Deepa Agashe in NCBS in Bangalore itself. She is also not my former student or postdoc. She was trained in the US.
Okay.
She does experimental evolution work using flower beetles of the genus Tribolium to look at many aspects of how organisms adapt to differing kinds of nutritional scenarios, and more importantly, what impact that has on their immune systems. Similar work on the interface of immunity and experimental evolution is also done by Dr. Imroz Khan in Ashoka University, Sonipat. So he does a lot of work with fruit flies and also with some other systems of insects, looking at how the life history of an organism, that is to say its rates of aging, its rates of development, its reproductive schedules, how they co-evolve along with the investment in immune function of different types, whether immune system evolves to invest more in tolerance of pathogen or resistance to pathogen, which are very different biological mechanisms. So what determines how an immune system evolves? That work he is doing. Dr. Shudipto Tung is looking at adaptation to different kinds of nutritional conditions in fruit flies over the long term. He is also in Ashoka University. Dr. Bodhisatta Nandi is in ISR Behrampur.
Okay.
He is working also to some extent on sexual selection, mate choice, and the evolution of these traits. So these are some of the contemporaries in India who are doing really good work in experimental evolution.
Here we are, signing off. Stay tuned with us to catch up on the latest science updates and learn about STEM experts